Hi everyone, we're going to go over our pre-lab material kind of in this video real quick. Hopefully it'll smooth things out for tomorrow. I just want to go over how microscopy works. So as we're viewing these cells, we need to use microscopes obviously because cells are too small for us to see with our naked eye. But we need to have an understanding of what our expectations should be. And so we need to understand the basic mechanics of microscopy. So how light microscopy works is you have a light source, and this emits all the light in the visible spectrum, red, green, and blue light. It will pass through a sample, typically in a slide. Some of these lights are absorbed by the sample, some are transmitted through. So the red and blue are transmitted through, whereas the green has been absorbed. It will go through an objective, and the objective typically magnifies. It's usually between 1, 5, 10, and 40x magnification. And then it will pass through the eyepiece, which typically also magnifies about 10x, before we view it in our eyes. Most light microscopy is going to be some variation on this basic schematic. All we really need to understand is it's based on light, which is a wavelength that is composed of many different colors. Some will be absorbed in the sample, and some will be transmitted through. And it's this absorption that actually allows us to see the sample. Now then, this is the stereotypical diagram of eukaryotic cell. I just want to flat out say it is actually incorrect. As you can see, there's no cell wall labeled, whereas there are clearly chloroplasts. I think they were trying to give you an idea of what you should expect to see in a cell. But because they've merged an animal and a plant cell, they got it a little wrong. But, I mean, they have the cell membrane, mitochondria, chloroplasts, endoplasmic reticulum, both smooth and rough here, ribosomes, which are all these little balls, nucleus, Golgi, and then the middle bit, which is the cytoplasm. So, this is your stereotypical cell. Why I wanted to go over microscopy and cells before we actually do microscopy to look at cells is I want to kind of set our expectations. So the size bar down here is 10 micrometers, which is equivalent to 0.01 millimeters. If I were to tell you the wavelength of light is 0.0 micrometers, which is 0.0007 millimeters, do we expect to see ribosomes, endoplasmic reticulum, the bilayer on the cell membrane, and the nucleus? So, pause the video, come back with an answer, and then we'll move ahead. Okay, so the wavelength of your light or your particle will actually dictate the resolving power of your microscope. So how small a sample you're able to kind of elucidate and observe. And because we know the wavelength of light is between 400 and 700 nanometers, or 0.0007 millimeters, we know that we can't resolve below that, because as the light is moving in the wave, if you are having a particle that's much, much smaller than the wavelength, the odds of that light wave hitting it are infinitesimally small. And so you need something bigger or as big as the wavelength for it to hit, and for it to be absorbed, reflected, refracted, etc. And so if we were to go back and look at these organelles that we expect to be smaller than the wavelength of light, we would not expect to see them in a light microscope. And so do we expect to see ribosomes? No, those are way smaller than the wavelength of light. Do we expect to see the endoplasmic reticulum? Probably not, because it's basically membrane that's just kind of there with nothing much in between, in the middle. So we don't expect to see that because the membranes are going to be much smaller than the wavelength of light. The bilayer of the cell membrane, no, that's way smaller than the wavelength of light. The nucleus, even though it looks really large, it's made up of a membrane, which is smaller than the wavelength of light, so it's thinner than the wavelength of light, and then DNA on the inside. And DNA is going to be much, much, much smaller than the wavelength of light. So, we can stain the DNA to make it easier to see, which will make the DNA larger than the wavelength of light. But generally, we do not observe nuclei in DNA when we look through light microscopes. Mitochondria, and this is once again an error in the diagram. They are actually typically pretty small. And here they're whopping big things, around 10 microns. Honestly, they're usually around half a micron. And so they're right at the resolving power of a light microscope. So sometimes you can if your species has large mitochondria, and sometimes you can't if they have small mitochondria. And then chloroplasts typically are larger than the wavelength of light. We should be able to see these with a light microscope. So I just want to set the stage for what we're going to do tomorrow, what we should expect to see, what we should expect not to see. And it all comes down to whether these organelles or components of the cell are larger or smaller than the wavelength of light. So I just wanted to kind of put that out there, that these diagrams tend to contain more information than what you'd observe. So tomorrow's lab, everyone knows that they're to either bring in a soil or water sample. Document, document, document. I want to know where you got it from, whether it was if it was a soil sample, if it was from your garden, if it was under mulch, if it was from your lawn, so just grass, if it was in an irrigation ditch, if it was at the side of water, if it was crud you find at the bottom of your pool. I want to know, because I want to know what we should. I want to know how much impact is man-made or human or anthropocentric. If you have a water sample, it could be from any water sample you'd expect to find microbes living in. So don't take a water sample from your faucet because it's chlorinated. I don't expect anything in there. So pond, lake, puddle, irrigation ditches are good too. And then just document where you got it from. Really, I want to know if it was a pond, lake, puddle, if it's in a residential area, agricultural, industrial. All this information will kind of help us determine whether we're having any impacts as humans on the microbial diversity around us. And so tomorrow we're going to kind of be looking at these under the microscope. We'll kind of be assessing species diversity, cell counts, and then hopefully we'll learn something as we go along. So we'll be filling out this tomorrow: sample type, collected from where, what kind of, if any, human impact there has been on the sample, number of living cell types, and then number of cells in total. So come tomorrow, be ready to kind of look at things under a microscope, and we will be doing some sketching and drawing as well. So bring your artist cap. So I look forward to it. See you all tomorrow.